Well, hello everybody, welcome back again to our next narrated presentation. Today we're going to talk about the group of organisms that probably represent the single most successful organisms that live on our planet and have probably ever lived on our planet. These are the prokaryotic cells, represented by bacteria and archaea. As the book very correctly points out right at the beginning of the chapter, these organisms are everywhere. If there is some unusual environment, some bizarre environment, some extreme environment, and there are organisms living there, they're going to be prokaryotes. In fact, it's really prokaryotes that define the boundaries of what is referred to as the biosphere, that part of the earth that is actually occupied by life. And this is because prokaryotes have this unique ability to adapt to the most harsh and unusual environments that you can imagine. So you can find prokaryotes, and particularly archaea, not so much bacteria, but very much archaea, living in environments that really stretch the limits of what life can tolerate. So extremely high temperatures. Bacteria can survive to temperatures in the order of 90 to 95 degrees Celsius, but there are archaea that can survive to temperatures above that, approaching 140 degrees Celsius. There are bacteria that can tolerate high salt concentrations, but there are archaea that can tolerate salt concentrations 10 times that of the ocean, basically almost pure sodium chloride. And there are bacteria that tolerate or require oxygen-free environments, anaerobic bacteria, but there are archaea that have an absolute intolerance of any oxygen at all. And these are extreme anaerobic organisms. And I'll introduce you to these later in the presentation when we look at the phylogeny of prokaryotes. We are going to be concentrating mostly on bacteria, because bacteria are the organisms that we have the greatest amount of knowledge about, but the knowledge that we have about archaea is expanding all the time. And the diversity of archaea is almost as great as the diversity of bacteria. Now, one of the things that the textbook also seems to stress is this notion of archaea as extremophiles. One of the things that you really ought to recognize is that while it is true there are groups of archaea that do live in these very, very extreme environments, they're not limited to extreme environments. Archaea can tolerate environments similar to those of bacteria. There is a huge diversity of archaea, just as there is a huge diversity of bacteria. But the one advantage that archaea have over bacteria is the ability of some of them, at least, to tolerate environments that are even more extreme than the most extreme that we know that bacteria can tolerate. So this, again, reinforces this high degree of flexibility and adaptability that prokaryotes have. So let's get started on this. Hopefully this is not going to be an extremely long presentation, but we'll see how we go, because there's a lot of interesting things to talk about. And we'll encounter some things that I actually worked on when I first arrived here in the United States as a postdoctoral fellow in California. When we get to that, I'll talk a little bit more about that, and I'll try not to get too carried away, because there is some really cool stuff here associated with bacteria and archaea. Anyway, onwards and upwards. So as I said in my introduction, bacteria are, well, prokaryotes are pretty much everywhere. They really do define the limits of where life can exist. So they do really define the limits or the boundaries of the biosphere. They are present in extremely large numbers. And this is one of the reasons why we have to start thinking of them as being the most successful organisms on the planet. Think about it this way. For about the first 2.5 billion years of the history of life on the planet, it was all about prokaryotes. They were the only types of organisms on the planet. In terms of eukaryotes, those have only been around at most for about 1.5 billion years, and multicellular eukaryotes have only really been around for maybe 1 billion years, if you want to push the envelope a little bit. Then if you want to talk about animals, well, animals have really only started to emerge, or really only start to emerge in the fossil record around 600 million years ago. So if you want to talk about the history of life on our planet, you really have to talk about the history of prokaryotes. They were the first types of cells to emerge from the abiotic soup of chemicals. And since that time, given that they had essentially a unique access to every ecosystem, every habitat, every environment that you could imagine, and because of their huge biochemical flexibility, they have come to basically occupy every niche that you can imagine. Now, what does limit them is the presence of liquid water or absence of liquid water. So they are still, like most life forms, dependent upon the presence of liquid water. That we still cannot deny. But what is interesting is that in some habitats, it is their activity that actually creates the liquid water. So one of the places that we do find prokaryotes that seems rather unusual is in amongst the ice of glaciers, and you have to go deep into the glaciers to really get an impression of how this comes about. But if you walk into the caverns that exist in the base of glaciers, and you walk in over the rock that the glaciers are traveling over, you can go hundreds of meters deep into the core of the glacier. And if you drill into the wall of the ice, you'll find little pockets of water. And in those little pockets of water, you'll find prokaryotes, bacteria specifically. And it is the bacterial activity that's actually creating these little pockets of liquid water that they can occupy. So yes, the prokaryotes can occupy all sorts of extreme environments, but this limit on requiring liquid water still exists. But in some cases, at least, it is their very activity that allows for the presence of liquid water that allows them to survive there in the first place. Going to the other extreme, if you go to the very depths of the ocean and find the deep ocean volcanic vents, what we refer to as black smokers, there you find archaea this time, a group of archaea known as methanogens, and we'll talk about them a little bit later on, that are tapping into the energy of chemicals in the hot water that's flowing out of these smokers. Now, the extreme vents have water in the range of 450 to 600 degrees Celsius. There are no organisms on the planet that can tolerate that at all, zero. But the water surrounding these vents is near freezing. It's around 4 degrees Celsius. And so the water cools down very quickly. And once it gets down to a temperature of around 140 degrees Celsius, maybe a little bit lower than that, you actually find archaea starting to occupy that water. And there are vents that are much lower in temperature than that, but these are the highest recorded temperatures that we found archaea living in. And they form the basis for ecosystems that have developed around these smokers, because they tap into the chemical energy through a process called chemosynthesis, which very much parallels photosynthesis, but it uses chemical energy, and specifically H2S and other similar sorts of chemicals as sources of high energy electrons to reduce carbon dioxide to organic molecules. So they become the basis for whole ecosystems in much the same way as plants are the basis for most terrestrial ecosystems, or most ecosystems on the planet, because of their ability to capture light energy. Here, the prokaryotes are capturing chemical energy and performing a chemical reaction that looks very much the same as photosynthesis. And part of our understanding photosynthesis was to come to an understanding of chemosynthesis first, back in the 1930s. which gave us clues as to how photosynthesis truly operated. Hopefully you learned a little bit about that in your Bio 1 class. If you didn't, not a big deal, but it is again a reflection of just how dependent life on the planet is on the function of prokaryotes. In fact, I'll put it this way. If the prokaryotes were to disappear tonight, the rest of life would disappear very quickly thereafter. As you'll see, there are some very central chemical processes that are carried out strictly by prokaryotes. And as a consequence of that, if the prokaryotes disappeared, the rest of life would disappear very quickly because they would it would very rapidly use up what remains of the prokaryotic product. And I'm being very careful not to say what it is right now because I want to introduce it to you a little bit later. But once that product disappears, the rest of life would collapse pretty quickly thereafter. So if we want to think about success of organisms, we think about prokaryotes. And we'll address this again a little bit later in this presentation, because there are a few other things that also point to why we need to consider them to be most successful, but I want to wait until a little bit later in the chapter to summarize a whole lot of things that I'm going to introduce you to along the way so that you can get a better feel for why prokaryotes truly are as successful as they are. So this is just another example of the flexibility of prokaryotes. These are archaea growing in salt water, and in fact this is saltier than seawater. It is seawater that's slowly evaporating, hence you can see the salt crystals around the margins of the lake. So the salt concentration here is quite high. It's several fold above that of seawater, and the way I can tell is because the water is turning pink and purple, and this is evidence of a group of organisms called halophiles. These are archaea and to some degree bacteria that can tolerate higher than seawater levels of salt. Archaea can take it to the extreme. Bacteria can still tolerate this. These are actually halobacteria. They have a pinkish purple pigment called bacteriorhodopsin that allows them to capture light energy and from that generate ATP. But they do so in very salty water. And so one of the indications that you get of increasing salt concentration above the levels of salt in seawater is the appearance of these bacteria and the changing color of the water. In places where they still collect salt by evaporating seawater, you find that the salt pans go through a series of color changes as different prokaryotes are able to colonize the salt pan on the basis of the salt concentration. So as the water changes color, as different prokaryotes colonize, then disappear and others take over, gives a rough indication of what the salt concentration is in the water, because each type of prokaryote that appears over time is comfortable with a particular salt concentration. That's when they colonize the salt pan, they get killed off as the salt concentration increases, but other prokaryotes then move in to take over. So it's kind of cool. It's an interesting observation. So let's take a look at the features of prokaryotes that allow them to be as successful as they are. So one of the things that hopefully you remember from Bio 1 is that all prokaryotic cells come in one of three shapes. Cocci, which are spherical, you see them here. Bacilli, which are rod-shaped, and you can see those here. And then spiral or corkscrew shape, which are the spirochetes and the spirilla. And while this may not look like a spiral, it's only looks like a wave because you're looking out from the side, but it is in fact a three-dimensional spiral. And many times the shapes are incorporated into the names of the bacteria. So if you did your Bio 1 class on West Campus, you probably depending on whether you did a wet lab or you did the virtual lab, but even in the virtual labs, you probably did this as well. You would have looked at bacteria that are responsible for making yogurt. If you didn't, not a big deal. But there are two types of bacteria that are responsible for making yogurt. They work together. One is called a lactobacillus. So we know that it is rod-shaped. The lacto tells us that it likes to metabolize milk sugar. And so it is one of the two bacteria that starts the souring process of the milk that converts it into yogurt. The other bacterium is a diplococcus. So the coccus tells us that it is ball-shaped or spherical, and the diplo simply refers to the fact that they like to get around in pairs. So when you take a look at a thin smear of yogurt under a light microscope, you can see the lactobacilli as short chains of rods, and you can see the diplococci as pairs of balls moving around together. So the sorts of things, the names often tell you something about the shape or include a reference to the shape of the bacteria. Not always. That's not always the case, but in a lot of cases that is encapsulated into the name of bacteria, certainly in some of the more modern strains that have been identified. When we start looking at the makeup of the cell itself, like any cell, it has a cell membrane or a plasma membrane, but outside of that cell membrane, like many cell types, there is a cell wall. In fact, one of the things that is going to become apparent as we move through this class is that animal cells, in many senses, are the odd cells out. And this is another way in which we can recognize that animal cells are an odd type of cell, the odd ones out, in that they are the only major type of cell that doesn't have a cell wall. Bacteria and archaea have them. We know plants have them, of course. Fungi have them. Many protists have them. But in terms of not having a cell wall, animal cells are the odd ones out. They are the one major line of cells that don't have a cell wall. So bacterial cells do have a cell wall, and it serves the same purpose as a plant cell wall, to stop the cell from swelling and bursting because it lives in a hypotonic environment. The water has a tendency to flow in, the cell would swell, the cell wall prevents that from happening. And one of the things that those of you who are planning to go into the health sciences, so if you're planning to go to medical school, pharmacy school, vet school, dental school, those sorts of careers should be aware of is that cell wall composition allows us to divide a lot of bacteria into two major groups on the basis of a staining technique called the Gram stain. The Gram stain is a two-part staining technique, and in between the two staining steps, you wash with ethanol, and depending on the chemical nature of the cell wall, either the original stain is retained and not washed out, or the original stain is washed out and you see the cells stained with the second stain. So let's take a look at what leads to that. So here are the two cell wall types. You have a cell wall that sits outside of the plasma membrane. So here's our plasma membrane, here's the cell wall. It's made out of a material called peptidoglycan. So this is a material that's made up of cross-linked polypeptides and sugars. And it is formed by enzyme activity. And the presence of that cell wall laying over the top of the plasma membrane shields the proteins in the plasma membrane from your body's immune system. And as a consequence, these cells are not identified by the immune system as being foreign invaders. And so the antibiotics that work against these cells block the activity of the enzymes that cross-link the polypeptides and the sugars to form the peptidoglycan. That leads to the loss of the cell wall, which exposes the proteins in the plasma membrane to the immune system. The immune system identifies them as foreign invaders and deals with them. So the antibiotics that are used against these cells do not kill the cells. They simply block the formation of the cell wall so that the immune system can identify them. The alternative type of cell wall does contain a thin layer of peptidoglycan, but the majority of the cell wall is made up of material called lipopolysaccharide. These lipopolysaccharides are extremely dangerous toxins. Some of the most potent bacterially derived toxins are these lipopolysaccharides that are present in this membranous layer that is part of the cell wall. And antibiotics that shut down the formation of peptidoglycan simply have no effect on these bacteria. And it's the makeup of these cell walls that determines whether they are identified as gram-positive or gram-negative. And being able to determine whether they're gram-positive or gram-negative tells you about the nature of their cell wall and tells you which type of antibiotics to start using against them, because using the wrong type of antibiotic simply has no effect. So a gram-positive bacterium traps the first staining component, which is a crystal violet. It has an intense purple color. So any cells that are gram-positive retain the crystal violet. Because of its intense purple color, you never see the safranin, which is a pink color. And so gram-positives stain purple. They have the peptidoglycan cell wall. And so these are the ones that are very sensitive to antibiotics that block the formation of peptidoglycan. Gram-negatives have the lipopolysaccharide component in this cell wall. There you can wash out the crystal violet. And so when you stain with the safranin, they stain a pinkish red color. And so you can see them here in the picture. It would be pointless using an enzyme that interferes with the formation of peptidoglycan here, because there's no peptidoglycan to lose in order to expose the proteins of the cell membrane to the immune system. So you need an antibiotic that literally kills the cell, that actually is toxic to the cell itself, the cell biochemistry. It would be pointless, however, to use those against bacteria that have a peptidoglycan wall. Could it kill the cells? Yes, but it is a needless use of an antibiotic that does not need to be used when these are sensitive to antibiotics that block the formation of the cell wall and allows them to be dealt with by the immune system. Because one of the other things that we also need to be sensitive to is the fact that bacteria are constantly developing antibiotic resistance. And the less we expose bacteria to antibiotics and needlessly expose bacteria to antibiotics, the more likely it is that we can delay the onset of antibiotic resistance to some of our more powerful antibiotics, which literally kill the cells. Again, we have to think in terms of, is it essential to use this particular antibiotic against this bacterium, or is there a better choice that would allow us to treat these bacteria without having to use some of the more exotic or more powerful antibiotics that we want to save for those bacterial infections where we really need them. So a simple test, usually carried out in a hospital med tech lab, determines whether you want to use an antibiotic that is targeted against the peptidoglycan layer, or whether you need to choose an antibiotic that is actually going to kill the cells by interfering with its biochemistry in some fashion. It's all a reflection of the cell wall. Then if we look around the outside of prokaryotes that like to stick to surfaces, we're going to find one of two ways for the bacteria to stick to the surfaces. Some bacteria have outside of the cell wall a polysaccharide. We actually refer to it as a mucopolysaccharide material. Sounds kind of disgusting, but it's basically a substance made out of long chain polysaccharides. It's sticky and allows them to stick to the surface of the teeth, or in this case, to the surface of the tonsil. This would be a strep infection of the tonsils. And you can see the tonsil material here and the bacterium is sticking to it. The alternative way is for them to use these extensions of the cell membrane called fimbriae. And this also allows them to stick to surfaces. So one of the things that you're going to see a little bit later in the presentation is an electron micrograph of something called a biofilm. And the biofilm that we're going to look at specifically is dental plaque. And dental plaque starts by some of the bacteria that live in your mouth, and you have more bacteria living in your mouth than there are people living on the surface of the earth right now. Yeah, it's that bad. And you can't get rid of them, nor do you really want to get rid of them, because they are protecting you against even more dangerous bacteria that are living outside of your body that would like to get in, but because of the bacteria in your mouth, there's no place for them to live. So we want them there. But you know that if you fail to brush your teeth for a little while, you start to get dental plaque building up on your teeth, and it begins by bacteria sticking to your teeth. That in turn allows dried saliva and food particles to stick to this mass of bacteria that is sticking to your teeth. And you start to get this thing called a biofilm building up on your teeth. It sounds pretty disgusting, and when you see the electron micrograph, you'll see it actually looks pretty disgusting too. More to come. One of the other things that we know about bacteria is that about 50% of all bacteria have a mechanism for moving through their environment. They can either directly swim using flagella that are of a different form to the flagella that you see in eukaryotes. There are some people that feel that perhaps it is bacterial flagella that may in some way have served as a precursor to eukaryotic flagella, but the structural and chemical differences between the flagella and the way in which they function suggests that perhaps this may not be the case, but there is still a hypothesis out there that suggests this. There's no direct evidence to support this, but nonetheless, that hypothesis is out there. So the ability to move, and let me pull up the pen. So the ability to move is called taxis. And it can be positive or negative. So those are the shorthands that I use for positive or negative. You'll see me use this rather than try and write it out in full. And so when we start thinking in terms of taxis, we then think in terms of what the stimuli are that are promoting the taxis. So you've got oxygen. Make it an N, which is called aerotaxis. And as you might guess, because there are anaerobes and aerobes, you can have negative and positive aerotaxis. Chemicals, and it can be a multitude of different types of chemicals. This is called chemotaxis. And again, it can be positive or negative. A more interesting one is light, and that's called phototaxis. And you might think, well, probably because of the role of light in something like photosynthesis would mean that you'd only have positive phototaxis, but in fact you can have negative phototaxis as well. Gravity. Yes, bacteria have the ability to detect gravity, and this is called gravitotaxis. And then one of the more interesting ones is magnetic field. This came as a real surprise, and this is called magnetotaxis. So apparently some bacteria are able to sense the Earth's magnetic field. We're not entirely sure why it's an advantage to them, but apparently they can respond to the Earth's magnetic field, because we know that they can respond to artificial magnetic fields in the laboratory. Now, this taxis movement gives rise to a way of moving through the environment called a random walk. So it's a random pattern. This box off. Oops. That we'll move to this one in a moment. So it's a random pattern. Called the random walk. So what's the big deal about this? Well, this is where my first postdoctoral fellowship comes into play, because when I first came to the United States, I worked in a microbiology lab where they were investigating aerotaxis. And so I learned a lot about bacterial movement through the environment. And the random walk is kind of an interesting thing. Bacterium will swim in a straight line, detecting what it can find in the environment. If it finds nothing after a period of time, it then tumbles in 3 dimensions, because it's in 3-dimensional space, and then heads off in a random direction. If it doesn't find anything going in that direction, then it tumbles again and heads off in another direction. If it discovers something that's of benefit to it, or obviously if it discovers something that it doesn't like, it changes its behavior. If it's something that it likes, it continues to swim in a straight line so long as the concentration continues to rise. Once the concentration starts to fall again, then it tumbles and swims off in another random direction. If it's something that it doesn't like, it immediately tumbles and moves off in another direction. And it is truly random. So you would think, if it's just swum through a concentration of material that it likes, surely it would tumble and swim back again. No, it doesn't. It just randomly tumbles and moves off in another direction. And the reason why it's random and it does this tumbling is because of the nature of the flagella that are driving this motion. So at the base of the flagellum, anchored in both the cell membrane and the cell wall, is this complex of proteins that make up a little electric motor, and it truly is a little electric motor. It is driven by the flow of hydrogen ions down through the motor. For some reason, I can't get the horizontal line. So I'm going to rub that out. Pen. So it's the flow of hydrogen ions, which is a flow of electrical current. Hydrogen ions have charges across this motor that allows it to spin. And because I had a lot of training as an electron microscopist and did a lot of work of using an electron microscope as part of my PhD, one of the first projects that I got involved with was growing up E. coli that had been mutated to lose their aerotaxis activity, and take a look at the motor to see if the mutation was affecting the motor at all. And it involves growing up huge quantities of bacteria and then very, very carefully isolating the motors by dissolving away the membranes and the cell walls, and then very carefully collecting them in a centrifuge so that you can look at them under the electron microscope. And I have a whole bunch of images that look just like this flagella motor. And like most complex structures, we've talked about the eye before as a complex structure and how it might evolve as a functional unit. Obviously, it can't evolve all at once, but if it evolves in a stepwise fashion, then you can develop over time through evolution's tinkering something that starts out very simple but evolves into something way more complex, so long as each of the intermediate steps is of benefit to the organism that possesses it. A similar sort of argument has been made for the evolution of this very, very complex little electric motor that motile bacteria possess, and it is very highly conserved across the motile bacteria. In fact, some motile bacteria lack the flagella. They don't have this filament region. What they have is the motor and the hook, and they can still move because they can embed the hook into a mucus material that they themselves are actually excreting onto the surface that they want to move across, and the action of the hook in the mucus will actually drive the cell forwards. So how does the action of this motor drive the cell forwards, and what causes it to tumble? Well, it turns out that the motor is reversible. When it spins in a counterclockwise direction, the flagellum or the multiple flagellum, if it has multiple flagella, bundle together to act as a propeller to drive the cell forwards. When it tumbles, the motor is simply reversed and spins in a clockwise direction. That, in the case of the bundle of flagella, causes the bundle to fling open, and that triggers the cell to tumble. In the case of the single flagellum, it interferes with the driving force forwards and causes the cell to tumble. Then once the cell has tumbled briefly, all the motors are switched back to counterclockwise rotation. The bundle comes together again and drives the cell forwards, and now it's moving off in another direction. Or that tumbling is suppressed and the motors continue to spin in a counterclockwise direction so long as it's sensing something that it likes and the concentration is increasing. So what sounds like a simple idea is actually quite a complex process, and a number of different sensors located in the bacterial cell membrane that are monitoring the different things that the bacterium likes or doesn't like are feeding signals to the motor to trigger either clockwise rotation or counterclockwise rotation. The full effect of all of these pathways feeding into the action of the motor is not fully understood, because obviously there are multiple signals that are triggering the motor to spin. At some point, one of them must override all the others to trigger the tumbling, depending on what the sensory apparatus is detecting and the signals that are sending to the motor. It's a very, very complex system, but nonetheless, a very cool system when you start thinking about it. So this is bacterial motility driven by these flagella. And just in case you were wondering what it's like for a bacterium to swim through water, it's kind of equivalent to us swimming through molasses. So you can imagine the sort of energy force that's being generated by these flagella. And there is a group of bacteria that are predatory bacteria that prey on other bacteria. And the way they do that is to swim at high speed and ram into their prey, burrowing through the cell wall. They don't penetrate the plasma membrane, but they do penetrate through the cell wall, and then they suck the guts out of the bacterium that they're preying on. And for them to penetrate through the cell wall, they have to swim at what would be the equivalent for us of swimming at around 60 kilometers an hour in molasses. Okay, so you start to get a feel for just how much energy is directed into these flagella and just how much force they can generate once you start to think about it in those terms. It's a pretty impressive system, no matter how you want to look at it. Once we start looking inside the cell. Once we get past the cell wall, the cell membrane, the capsule, we get inside the cell. Basically, what we find is a single compartment, although there are regions that are set aside for various functions. So if we look at an aerobic bacterium, one of the things that we find is these layers of membrane. If those look a lot like the Christian side of the mitochondrion, that's good, because this is probably where those Christian membranes came from, because remember, the origin of mitochondria is amongst the beta proteobacteria. These are aerobic bacteria, and we know that all aerobic bacteria have these membranes in which the electron transport pathway is located, the ATP synthases are located, and again, these membranes increase the amount of membrane area for the generation of much more energy. But they are all derived from the cell membrane. So if you look carefully here, you can see that, and over here as well, you can see that what looks like separate membranes actually are continuous with the plasma membrane. So these are infoldings, extensions of the plasma membrane to create a larger surface area for the benefit of carrying out aerobic respiration. Then if you look in cyanobacteria, again, you have this highly folded membrane system. This is where photosynthesis occurs. They are actually genuinely referred to as thylakoid membranes, just like the membranes inside the chloroplast. And again, very careful electron microscopy has indicated or demonstrates that these membranes are infoldings of the plasma membrane of the cell. They are not a separate membrane system. They are continuous with the plasma membrane. So these are infoldings of the plasma membrane to increase the surface area for carrying out these two very important biochemical pathways, cell respiration and photosynthesis. And remember again, chloroplast derived from cyanobacteria. Then another region that is specifically set aside inside bacteria is a region called the nucleoid. This is where you're going to find that copy of the circular DNA molecule that represents the bacterial genome. Now, I know the authors of our textbook have adopted the use of the term chromosome for this circular DNA molecule. And I've noticed that amongst authors of freshman biology textbooks, this is a fairly common thing, in part because once one group of authors does it, everyone else basically does the same thing too. I'm not terribly comfortable with using the term chromosome for a circular DNA molecule by virtue of the fact that historically the term chromosome is referred to the linear DNA molecules that you find inside the nucleus of eukaryotes. I'm more comfortable with using the term genome. But given that the book uses the term chromosome, if that's the term that you want to use and want to apply here, that's fine with me. Just understand that when you go on to your more advanced biology classes, you're probably going to find that the term chromosome is not widely used unless you encounter someone who has decided to adopt that term, because the more traditional term for this would be genome, chromosome reserved for the linear molecules. So this large accumulation of DNA, this is that single circular DNA molecule that has exploded out of this E. coli cell. So this is a single E. coli. It was contained within the nucleoid region inside the cell. So you can imagine that it was very highly packaged to fit inside the cell, not just inside the cell proper, but within a unique region inside the cell. So there is a degree of DNA packaging that goes on here, but it does not involve a unique group of proteins like you find in a eukaryote. You may recall the histone proteins that are used to start packaging the chromosomes in the nucleus. We don't find those sorts of proteins here in prokaryotes, but there is obviously a large degree of packaging. It just doesn't involve specialized proteins to do the packaging. But then you also need to recognize, and I'm going to change the color here. I like the violet, but there are a second group of circular molecules that are much smaller. These are referred to as plasmids, and the Reason why plasmids are important is because these are the primary pieces of DNA that bacteria exchange with each other. And there are, in fact, a number of different types of plasmids. There's an F plasmid that we'll talk about that triggers the formation of a structure that allows for direct cell-to-cell exchange of DNA. There are other plasmids that contain helper genes, and also there are some special plasmids that are now being referred to as R plasmids, because these seem to be the location of the vast majority of genes that give antibiotic resistance to the bacteria that contain them. And this is one of the reasons why antibiotic resistance has become such an issue, because these plasmids are not just exchanged between bacteria of the same species, they can be exchanged between bacteria of different species. And so species of bacteria that have never encountered an antibiotic can possess a resistance gene to it because they've got that gene from a bacterium that previously had an antibiotic or spontaneously developed an antibiotic resistance to that particular antibiotic, encapsulated the gene for it in a plasmid, and as they grew in numbers, because the antibiotic was killing off all of those that didn't have the plasmid, they left the plasmid around available for other bacteria to take up, which they will do. Now, in microbiology labs, in genetic engineering labs, we actually use the ability of bacteria to take up plasmids to engineer bacteria by inserting genes into plasmids that we want to insert into the bacteria, and then we trick the bacteria into taking up the plasmids. You could just insert the plasmids into the solution of bacteria and they would spontaneously take them up over time. But you can increase the likelihood of them taking up the plasmids by carrying out a process called heat shock. You take the bacteria in a tube, you place it in 40-degree water. It's not hot enough to kill the bacteria, but it loosens up their cell membranes and increases their permeability to the plasmids. That increases the likelihood of the plasmids entering into the bacteria, and you only need to do this for about 30 seconds. And then you cool them back down very rapidly by plunging them into ice. That tightens up the membranes. And so in that short exposure time to the solution containing the plasmids will have entered into the bacteria. By tightening up the cell membrane again, you lock the plasmids inside the cell, and then you can use a number of different ways of identifying which bacteria have taken up the plasmid or not. Today, we use fluorescent proteins that have been obtained from deep water jellyfish as markers to identify which cells have taken up the plasmid and therefore likely also taken up the genes of interest that we're really after. We just use the jellyfish proteins as a marker to indicate whether we've been successful or not. In the past, I've used antibiotic resistance. Yeah, I know that doesn't sound really smart, but at the time, it was all we had. Subsequently, we got away from that as we realized it was not a smart thing to do and used a substance that is a substrate to the same enzyme that's used to break down lactose sugar. And it turns the bacterial colonies blue in the presence of this alternative substrate. And now we use the fluorescent proteins, the fluorescent jellyfish proteins as markers. So we've come a long way in identifying markers. And just in case you're curious, we have 3 jellyfish proteins. The first one was called green fluorescent protein, or is called green fluorescent protein, otherwise known as GFP. Then we have a red one, which is very cleverly called red fluorescent protein. And we have a yellow one, again, very cleverly called yellow fluorescent protein. And these are the marker proteins now that are used in a lot of plasmids. And you can actually buy plasmids that have been engineered by molecular biology firms for the purpose of inserting genes into bacteria. And they have known sequences of nucleotides that you can leverage to insert your genes into the plasmids. We don't really have time to talk about how that is done in this presentation, but maybe at some point we can talk about that if we have time, sometime in the future. Another advantage for prokaryotes is simply the fact that they reproduce very, very quickly. Now, on average, bacteria will divide somewhere around every hour or so. In less than ideal conditions, it could be 3 hours. In some bacteria that we've discovered buried in little pockets of water in rocks that are several kilometers below the surface of the earth, at the bottom of some of the deepest mines on the planet. Those bacteria are thought to divide roughly about once every 1,000 years or so, simply because there's very little energy for them to utilize. They're dependent upon minerals that have been solubilized out of the rocks. So they don't have access to a whole lot of ATP, but they do have enough to keep them alive, but that's about all. So they only reproduce about once every 1,000 years or so. That's best estimate. So there's a wide variety of replication times. In laboratory conditions, E. coli, the bacterium that lives in your colon, your lower intestinal tract, and is used for a lot of molecular biology work in microbiology labs and elsewhere, they can reproduce as quickly as 20 to 30 minutes under ideal conditions, but that's laboratory conditions where you've provided them with everything that they need in ideal conditions. 37 degrees Celsius, all the metabolic resources that they need to grow as quickly as they can. But under most conditions, they don't grow that rapidly. If things turn really bad, and by really bad, generally we mean water starts to become a limiting resource. Many bacteria have an interesting way of protecting themselves. They form an endospore. So here you can see the original bacterial cell. And then you can see this rather dense structure in the center. That's where the bacterial genome is located, along with support material that will allow this to be recovered once the cell encounters, or the endospore encounters viable conditions. And it coats this region with a very hydrophobic coat that prevents it from dehydrating. And these can sit stable in the environment for long periods of time. Now, one of the bacteria that are notorious for doing this is the anthrax bacterium. In fact, about a year after the 9/11 attacks, we had a couple of outbreaks of anthrax, one in Boca Raton here in Florida, and a couple of instances in Washington where letters were found with white granular powder in it. Unfortunately, in Boca Raton, several people got infected with anthrax as a consequence of this, and at least one person died. And that's because the anthrax, this white granular powder, is actually the endospores of the anthrax bacteria. And there are two forms of anthrax infection. There is the infection of the skin, and there's the infection of the lungs. And if it gets into the lungs, it's almost always fatal. And that's what happened to the unfortunate individual in Boca Raton. In Washington, no one got infected, fortunately, but several buildings, congressional buildings, had to be shut down as a consequence of receiving mail. This is one of the reasons why all the mail in the United States now is subject to X-ray by the Postal Service. Try and figure out what's in there. The anthrax that was used, somewhat ironically, was traced back to Fort Detrick, an army base near Washington in Maryland, where the army carries out its germ warfare research. And it's one of two locations where there are known samples of anthrax in captivity for use for study. The other is a laboratory in Russia, where they also carry out germ warfare research. These are also the only two locations that we know of now that still have viable smallpox virus particles. Again, for research, or at least that's the claim. And once it was recognized that the anthrax came from Fort Detrick, then the chase was on to try and figure out who actually was responsible. Eventually, the FBI pointed their finger at one of the scientists that had worked in the research facility where the anthrax was kept. They could never say for certain that this individual was responsible, but they made public that this was their suspicion, and the weight of public scrutiny after that apparently bore down on this individual so strongly that eventually he committed suicide. We don't know for sure whether he was responsible, but this is certainly the individual that the FBI pointed their finger at. So what's the point of talking about anthrax? Well, it turns out that this is not a new situation. Anthrax has been played with before as a potential biological warfare agent. So in World War II, both the Germans and the British experimented with it, and the British did their experiments on an island off the coast of Scotland, if I remember rightly. It's several miles off the coast, but this island is completely contaminated with anthrax spores, so much so that periodically, when dust blows to the mainland, there's an outbreak of anthrax because the spores get transported to the mainland and you get an outbreak of anthrax. And there's no way of eliminating the spores. They're very stable, they're in the dust. The British have tried sealing over the most contaminated areas with asphalt, but spores still escape. And they are obviously still viable nearly 100 years after, well, 80 years after the end of the war, because every time that they get carried to the mainland, there's an outbreak of anthrax. These are still viable spores. We don't know how long these spores are going to remain viable. This is kind of, unfortunately, an ongoing experiment in just how viable these endospores are. So anthrax is not the only bacterium that can form endospores. There are lots of bacteria that form endospores. And again, those of you who are going into the health sciences, you're going to have to deal with this. And you'll learn more about this in the microbiology courses that you'll take, either in your 4-year degree or once you enter medical school or whichever program you're going to go into. So let's take a break. We've been going for a while. Here's our concept check question. I am going to see if I can't get this to break the recording and come back again. So I'm actually going to physically stop the recording and we'll see how we get on with this. So here are my 2 concept check questions for this break. The success of prokaryotes is due in part for their ability to adapt to environments too harsh for other organisms. Identify 2 adaptations that allow for this. And how do cellular and DNA structures of prokaryotes and eukaryotes differ? I'll see you on the other side of the break with the answers.